Thank you very much for the nice introduction and for giving me the chance to present this work. I'm Fangyun. I'm an instructor working in Dr. Patrick Peled's lab. Over the past few years, we have been working on studying the neural circuit mechanisms of different anesthetic drugs, including ketamine and propofol, using intracranial recordings in human patients. As an anesthesia researcher, we are interested in understanding how anesthetic drugs work and to identify biomarkers to measure brain states clinically so that we can ensure that our patients are receiving the right amount of sedative or anesthetic drugs. And the primary tool we use to measure brain states is EEG. We collected raw EEG signals from the brain, like the traces shown in panel A. Then we use signal processing method to convert these raw signals into this colored map called a spectrogram in panel B. With the y-axis shows the frequency and x-axis shows the time. The spectrogram can tell us which frequency components exist in the raw EEG signals. In this way, we can track the changes of brain dynamics over time on the different drugs. We could see these 4 common anesthetic drugs, they produce very different EEG spectrogram. These EEG signatures can be related to their different molecular targets and the neural circuits, like propofol produce the slow and alpha oscillations, mainly through the GABAergic inhibition mechanism, whereas ketamine produces the high-frequency gamma oscillation, mainly through the NMDA inhibition mechanism. Ketamine is of great interest to us because of its unique properties. Different from other anesthetic drugs, which are mostly associated with low-frequency EEG patterns, ketamine produces high-frequency oscillations that are often considered as indicators of a highly activated brain, even though the patients may be unconscious. Ketamine's effects are also dose-dependent. At anesthetic doses, ketamine could induce loss of consciousness for general anesthesia, but at the sub-anesthetic doses, ketamine could produce dissociative states. In addition, sub-anesthetic doses of ketamine could also induce significant and rapid antidepressant-like response in treatment-resistant depressed patients. However, the neuropsychiatric side effects has limited the extensive use of ketamine in clinical practice. If we could define the neural circuits engaged in ketamine's antidepressant and dissociative effects, that would help with the development of improved therapies with fewer side effects and greater safety. Ketamine is widely known as an NMDA receptor antagonist. At sub-anesthetic doses, ketamine blocks the NMDA receptors on GABAergic inhibitory interneurons. This results in disinhibition of the downstream excitatory pyramidal neurons. In addition, ketamine has also been identified as HCN1 inhibitor. The HCN1 channels are concentrated in cortical and hippocampal pyramidal neurons, where they play an important role in determining synaptic input integration and the neuronal output. There are increasing evidence suggesting that the HCN1 channels may be involved in ketamine's dissociative and antidepressant effects. The research questions we were interested in include what are the molecular targets and the neural circuit mechanisms underlying ketamine's antidepressant and dissociative effect. If we could collect EEG signals before and after ketamine infusion, we would be able to characterize the oscillatory power changes induced by ketamine and study how these oscillatory power changes relate to the neural circuit mechanisms underlying ketamine's antidepressant and the dissociative effects. We could also be able to characterize which cortical and subcortical structures play a major role in mediating this process. Before jumping into our study, I would like to briefly talk about two other EEG studies which investigated the mechanism of subanesthetic doses of ketamine-induced dissociation. One is this paper published by Willis in 2018, in which they showed that the reduction of alpha power at the precuneus and the temporal parietal junction is the mechanism for subanesthetic ketamine-induced dissociation. They compared EEG power changes before and after ketamine infusion in healthy subjects. Figure on the left shows the power during baseline in blue color and after ketamine infusion in red color. X-axis shows the frequency and the Y-axis shows power. They found that after ketamine infusion, there was a global decrease in low-frequency power. Figure on the right is the quantification of a figure on the left. They summarize the power changes at 5 frequency band and 4 channel clusters, anterior, central, parietal, and occipital. Negative values represent decrease in power compared with baseline, and the positive values represent increase in power. We could see that the power increase was only observed in the gamma frequency. And the power was reduced throughout all low frequency channel clusters. The largest reduction is the alpha oscillation in parietal and occipital channel clusters. In order to find out the localization of the source for these power changes captured with the surface EEG, they conducted the EEG source estimation analysis. They identified significant effects in source estimates for only alpha oscillations. Figure A shows 3 different views of the source estimation results. They found significant decrease in 22 voxels, mainly at the right lateralized temporal parietal junction and the superior parietal regions. Significant voxels were then reduced into 4 small non-overlapping clusters: the medial temporal gyrus, inferior parietal lobe, supramarginal gyrus, and the precuneus. The conclusion they made for this study is that ketamine induces altered states of consciousness during periods of reduced alpha power in the precuneus and the temporal parietal junction, and the modulation of these temporal parietal loci are the candidate mechanisms for the psychoactive effects of ketamine, given that these regions is involved in multisensory integration, body representation, and consciousness. Interestingly, in 2020, there was this Nature paper published which provided evidence for the role of deep posterior medial cortical rhythm in dissociation. The posterior medial cortex, or PMC, in human is defined as the retrospinal posterior cingulate and the medial ventral precuneus cortex, which constitute a core node of human default mode network that is known to be engaged during internally focused subjective cognition, such as autobiographical memory. The first of their study was conducted in mice that I will not talk in today's presentation, but from the mice study, they found that subanesthetic doses of ketamine could induce a 1 to 3 Hz rhythm that only exists in the mice PMC and may be related to the dissociative experience. To test this 1 to 3 Hz PMC rhythm, if it exists in humans, they recorded the electrical activity from multiple brain regions in one human patient with epilepsy. This patient has epilepsy with a pre-seizure aura that were described as dissociative. This figure shows 10 seconds intracranial EEG traces from this patient near the onset of seizures with a dissociative aura. We could see that the prominent rhythmic waveform only existed in the deep PMC on both hemispheres. With the power spectral analysis, they found a sharp oscillatory patterns centered at 3.4 Hz that was restricted to the left and the right deep PMC, but not the superficial PMC or any other brain regions. As part of the brain clinical mapping, the brief electrical stimulation was applied to individual electrodes. They found that stimulation of the seizure focus in the right PMC could induce dissociative aura-like feelings, similar to those seizure onset, and the stimulation of the left PMC induced immediate and confidently reported dissociative experience without the feeling of an impending seizure. The author thinks these data suggest a causal relationship between human dissociative experiences and the 3.4 Hz rhythm in the deep PMC. Here is a summary of the two studies. From the 2018 study, we learned that the increase of gamma power and the decrease of low frequency power are the EEG signatures of subanesthetic ketamine-induced dissociative experience. And the alpha power reduction in precuneus and the temporal parietal junction may be the potential mechanism for ketamine-induced dissociation. The 2020 study has the advantages of using intracranial recordings and electrical stimulation to establish a causal relationship between PMC rhythm and dissociation. Although they found a strong 3.4 Hz rhythm in one human subject that occurred during the epilepsy aura, the subject did not receive ketamine. So it's still unclear whether this 3 Hz PMC rhythm also exists in ketamine-induced dissociation. To characterize the brain dynamics during ketamine-induced dissociation, we collaborated with Dr. Sidney Cash's lab in neurology department at MGH. Their lab has the expertise of conducting intracranial EEG recordings with depth electrodes in patients with treatment-resistant epilepsy. These patients, they came to the hospital and have intracranial electrodes inserted into their brain for monitoring for like 1 to 2 weeks to identify the seizure onset zone. The figure on the right shows the distribution of the intracranial depth electrodes in a patient with temporal lobe epilepsy. With the brain mapping tools, we were able to localize the precise location of each electrode. This surgery setup gives us a great chance to study the brain dynamics occur in multiple cortical and subcortical regions before and after the drug administration. So we designed this study to measure these intracranial EEG activities in human patients who received a subanesthetic dose of ketamine before the induction of general anesthesia with a propofol for the electrode removal surgery. We recorded the baseline signals for 5 minutes, and then each patient received an infusion of a subanesthetic dose of ketamine. Total dose is 0.5 milligram per kilogram over 14 minutes. Patients then completed a questionnaire to evaluate dissociative states. Propofol bolus was then given to the patients to induce general anesthesia. Our goal was to characterize the brain regions involved in ketamine-induced rhythms in order to better understanding their potential role in mediating ketamine's dissociative and antidepressant properties. We also looked for evidence of the 3 Hz PMC rhythm identified in that Nature paper. At the neurocircuits level, we also studied the role of the molecular targets such as NMDA and HCN1 receptors in producing ketamine-induced oscillations. This is by analyzing the interactions between subanesthetic ketamine and propofol. Propofol is a GABA receptor agonist and HCN1 blocker. We expect that propofol's GABAergic activity would antagonize any ketamine-induced oscillation, ketamine-induced oscillations from the NMDA-mediated disinhibition. At the same time, we expect that propofol would further potentiate any ketamine-induced oscillations from HCN1 inhibition. We collected the data from 10 subjects, 5 male and 5 female, with age ranges from 22 to 59 years old, an average of 35.9. There are in total of 825 bipolar channels. We are using an abbreviation version of the questionnaire to assess the dissociative states. Example questions are like, do things seem to be real, unreal to you as if you are in a dream, or do colors seem to be diminished in intensity? Patients answer yes or no to the questions. The response on the questionnaire are consistent with the dissociative states induced by subanesthetic ketamine. I conducted a power spectral analysis. Here shows the power spectrogram for 3 representative channels. We could see that different channels showed distinct changes of EEG power after ketamine and propofol. Under ketamine, we observed increased gamma power in the inferior frontal and decreased alpha power in the middle, temporal, and occipital channels. After propofol was added, there was a large increase in power in the inferior frontal and the middle temporal channels for nearly all frequencies, except for the high gamma band. In contrast, the reduction of alpha oscillation in the occipital channels was further enhanced with the adding of propofol. This result suggests that the intracranial EEG dynamics induced by ketamine and propofol are location and frequency dependent. To obtain a comprehensive understanding of how these changes in brain dynamics in relation to the brain anatomy across all subjects, we analyzed the power spectral changes in regions of interest based on the brain atlas and plotted them against the template brain. The channels were assigned to structural labels based on cortical parcellation with the DKT-40 atlas. In this figure, warmer color represents increase in power. Cooler color represents decrease in power after ketamine. We could see that ketamine induced a global decrease of power at slow, delta, theta, alpha, and beta frequencies for most cortical and subcortical regions, with the largest reduction in occipital regions at alpha frequency. Consistent with the previous studies, a large increase of gamma power was detected in frontal regions of the brain. We did a same analysis for after the propofol. As what we expect, adding the propofol dramatically reversed the EEG power changes induced by ketamine. As you can see in the picture, from slow to beta frequencies, all structural regions showed a remarkable increase in power except for occipital regions, which displayed a further decrease of power at theta, alpha, and beta frequencies. For gamma frequencies, there is a large decrease of power in frontal regions and the increase in power in precentral, postcentral, temporal, and occipital cortices. This result suggests that propofol both enhance and antagonize EEG power changes induced by ketamine, and that effects are frequency and location dependent. Based on the above data, we proposed the possible neural circuit mechanisms for the changes in frontal gamma and occipital alpha power after ketamine and propofol. We found that ketamine increased gamma power, whereas propofol decreased gamma power in prefrontal regions of the brain. This may be explained by the antagonistic effects between ketamine and propofol. Ketamine preferentially blocks the NMDA receptors on the GABAergic inhibitory interneuron, resulting in inhibition of the downstream excitatory pyramidal neurons, which mediates the increased gamma activity. When a GABA agonist propofol was added, it antagonized the gamma power increase, possibly by restoring some of the inhibitory activity in the affected brain regions. We also found that ketamine and propofol both decrease alpha power in the posterior sensory cortical regions. This may be because of the shared inhibition of ketamine and propofol on HCN1 channels. And this is supported by modeling study, which showed that reduction in the IH current mediated by HCN1 could abolish the occipital alpha by silencing thalamocortical cells. To further test the role of the 3 Hz rhythm in ketamine-induced dissociation, we analyzed the spatial distribution of the 3 Hz rhythm after ketamine and propofol. We found a significant increase of 3 to 4 Hz power in posterior cingulate and isthmus cingulate regions, which are part of the PMC, as well as the pars opercularis region, which belongs to the inferior frontal cortex. We then ask if the power increase in PMC is centered at 3 to 4 Hz. By plotting the power differences for PMC as a function of the frequency, we found that the increase of 3 EEG power after ketamine peaked between 3 to 6 Hz. Here is the 3 to 4 Hz power changes after propofol. We found that propofol significantly increased the 3 to 4 Hz power in most of the brain regions, including the PMC, suggesting that the effects of ketamine and propofol on the 3 to 4 Hz PMC rhythm are additive rather than antagonistic. From this data, we found that ketamine and propofol both increase 3 to 4 Hz power in posterior cingulate and isthmus cingulate cortex. The neurocircuit mechanism was proposed as below. According to the Nature paper, they are both NMDA receptors and HCN1 channels in deep PMC, both of which are required for generating the 3 Hz activity. It's possible that ketamine blockade of the NMDA receptors could hyperpolarize the membrane potentials to in the PMC and activating the intrinsic HCN1 channels and produce the rhythmic dynamics. The same thing could be true for propofol by GABA-mediated hyperpolarization. Although both ketamine and propofol induced the 3 Hz rhythms in PMC, dissociation was only detected after ketamine. This may be because propofol suppresses arousal and induce unconsciousness, which would supersede any dissociative effects. This is just when I put all the mechanisms together. The limitation of this study is that we did not directly measure clinical depression or antidepressant effects. Future studies in depressed patients are needed. In this study, we focused primarily on the role of NMDA and HCN1 receptors. The role of other receptors that have been suggested to play a role in ketamine's antidepressant effects could also be investigated in the future. In follow-up studies, it would also be interesting to explore the relationship between EEG oscillatory dynamics and the intensity level of dissociation, which could not be addressed in the current study due to our limited sample size and the limited resolution of dissociation measurements. Cross-frequency coupling analysis could be an additional topic of interest for characterizing the interactions between oscillations at the different frequency bands. Overall, we found that ketamine has distinct dynamic effects on neural system known to mediate depression and sensory processing by multiple mechanisms. The neurocircuit mechanism underlying ketamine's induced oscillatory dynamics and their potential links to antidepressant and dissociative effects, as proposed in this study, may have important implications for the development of novel therapies with fewer side effects and greater safety. I would like to thank my mentor, Dr. Patrick Peled, all the collaborators and the study subjects and the funding agencies for their great support to the project. Thank you very much.
Thank you, Fangyun. That was really interesting, and thank you also for the background explanation and really like a nice story that what led to your project. Again, if you have any question, please just like raise your hand or just write them down in the chat. But while you're thinking, maybe in the meantime, I can start with a couple of questions on my own. I think it was really interesting, but I can see how it may be difficult to apply this kind of study in clinical practice because you are using intracranial electrodes. How would you see like translating this study, like for example, in a clinical population, right?
These are in clinical population because the intracranial recording can only be obtained with patients with epilepsy, but we can do high density EEG studies in patients with depression, and we are planning to do that with a collaboration with some psychiatry faculties at MGH to start that study. And with a combination of the MRI imaging, we will be able to do source localization so that we can localize the source of the surface EEG activities so that we know what happened within subcortical regions. I feel it's very translational. Yes. We would like to see if we can see these patterns with a surface EEG too.
And I noticed that like you were talking about like a 14 minutes, right, sub-anesthetic infusion that is actually shorter than what we do in clinical practice. I assume that was like due because of the surgery protocol, or was like
because of the limited time in the operating room, we don't want to delay the surgery procedure. And we did some simulation with a PK/PD model, and we could reach higher effect site concentration with this design, with this shorter time frame. We compared it with the regular 40 minutes administration regime, and these 14 minutes could reach a higher effective site concentration compared with the 40 minutes.
Do you also see, because these were a patient with like medical refractory epilepsy,
yes,
would like be would you expect this to be different in a population that does not suffer from that condition? Like, for example, like a depressed population that would not suffer from epilepsy, would the baseline recording be different? Or
I think, they will be different because there are some papers showing that the brain, the EEG dynamics in patients with depression are different from the normal subjects, and will also be different from patients with epilepsy. There is no consistent conclusion about what are the differences, because that depends on which brain regions are damaged in those patients, are affected in those patients, and the brain states. But they will be different, I will expect.
I think we have some question in the chat. I saw that like you posted some question. Would you like to ask them directly, or should I read them out for you?
Let me see. Open chat.
Hi.
Because
if those patients were screened for depression because it's a common comorbidity, epilepsy and depression, any kind of structurized clinical interview for depression on those patients?
I do check the history for those patients, and half of them have a history of depression, but they are not assessed clinically based on, so I cannot say they definitely have it when we do that experiment. But half of them do have a history of depression from their record.
And they were probably on medication during the recordings, right? With oral medication?
No, I don't think so. That's the only drug we give is the ketamine during our experiment.
Oral medications for epilepsy or for depression? On those medications?
No, because when they are doing the clinical monitoring for the epilepsy, they are off all the medications.
Okay.
Because they want to find out the location of the onset zone for the epilepsy.
And just one comment that even though propofol and ketamine have different opposite ECG signaling, both are antidepressant and both can produce antidepressant activity. It's very interesting to notice that.
Yeah.
Thank you.
Thank you for the questions.
Well, we have a question from Rupert.
About the sample size. Yeah, I feel that's the limitation of our study because of the sample size, and we did a abbreviated version of the questionnaire, so we are not able to do a grading of like assign a number to assess the degree of dissociation. So we cannot do that correlation analysis with our current data. So those.
Because I think in the way you use the cards, instead of like writing a score,
Yeah,
just like
yeah.
Yes, no variables, right? So
yeah, yeah. In the original version, they have like ranking from 1 to 5 to score how much your dissociation is for each question. But in our version, we just let them to ask yes or no because of the limited time in the OR.
But you could just sum the total of those CAD scores, and you'd have then a sort of just some sort of very basic correlation, or in fact you could dichotomize them between those that dissociated and those that didn't, and just explore whether there was a difference in the effect on the power and the degree of in the effect of the power, the change in power.
Yeah, that would be interesting.
I mean, it's highly exploratory, of course, but
yeah.
The reason that I think it might be worth just having a look is that if you've got obviously there are two big effects of ketamine. There's a reduction in alpha power and the increase in the gamma power. These are things that you might be able to measure really very easily, and you don't even have to use a high density sort of montage. You could use a very simple montage with four EEG electrodes, and if you could find that there was a sort of link between the alpha power reduction and the dissociation, then this might be helpful clinically.
Yeah.
Particularly if your intention is to titrate to the effect, as anesthetists do. They commonly titrate to a point where people go to sleep, but you could titrate to dissociation, and sometimes it's just a little bit difficult for people to report that because it's an unfamiliar experience. And so if you could titrate to an alpha power reduction, then that would be potentially useful, and particularly if people are getting repeated ketamine, you could have a look and see if that varied with time.
Yeah.
But obviously that go on,
because
sorry.
you are talking about the alpha power, and what we found is the 3 Hz PMC rhythm, maybe the mechanism for the dissociative experience, because the PMC rhythm is kind of deeper inside the brain. So I'm not sure if only like a few channels of EEG would be enough to capture this rhythm.
No, fair comment. But presumably is there a correlation between that and the alpha power?
No, that was identified by the 2018 study, and that is a little different from our study, the conclusion.
The conclusion's different.
Yeah.
Okay. Okay, thank you.
You're welcome.
I think we have another question. Luke, I don't know if you're here, if you want to ask it directly, or
you mean the question from Luke? Other drugs administered during.
So the question is, do you consider effects of other drugs administered during the anesthesia period, like fentanyl or midazolam?
Yes. Yeah, we do have those drugs administered together. And we are currently, fentanyl is a topic that our lab is studying too. Based on my experience, the EEG power changes for alpha is the most predominant over other power changes induced by other drugs like fentanyl. It's more subtle compared to the effects of the propofol. But we are studying the other drugs, the effects of other drugs on the power spectral changes. in other projects.
Thank you. Just a quick other question. With the participants, did they have their eyes closed or?
Yeah, they have eye closed during the whole process.
Okay, perfect. Thank you.
You're welcome.
And thank you. The effect that you have observed were like throughout the time of the administration of ketamine, right? But would you expect them to stay consistent throughout all the administration time, or would they vary?
During the baseline, they just are very consistent. They just have eye closed, and we do have some behavioral tasks where they listen to some auditory task and would press the mouse based on what they hear, so that we can track their conscious levels, and they can do that, still do that after the ketamine infusion. And when I give them questionnaires, they are all awake and are able to answer my questions. And then after you give them the propofol, they all lost the consciousness.
Right.
Yeah.
And I think, just like to tie up, I think through what Rupert was saying, is like, would you expect these to be a constant effect? Or let's say over time, if people receive more than one infusion, will this signal change? Yeah,
I will expect them to be changing. Yeah. Because these EEG dynamics are a real-time marker of what happens in the neuronal levels. If you give more ketamine, the brain power changes will increase. They will change. That's why we use them as a marker for the unconscious levels for anesthesiologists.
Yeah, but you were talking about like the total amount of ketamine they would be receiving. So if you change the total amount during one administration, then of course these changes would be different.
Yes, it will be different.
But I think our question is like, if you receive ketamine 1 week and then you receive another infusion the following week, do you think that same dose, same time, do you think that these signal, these neural changes that you see during the infusion would be different?
That I have no experience with that. Yeah, I'm not sure.